Hello everyone, welcome to the webinar on Bactericera cockerelli, one of the EU priority pests. As you might know, we have already provided 3 webinars on EFSA's plant pest survey methodological framework and the toolkit, including the repest and the OptiPest, and 4 webinars on fruit flies, Tephlididae, and 11 webinars on EU priority pests. If you did not attend the previous webinars, and you would like to know more about them, the recorded presentations are available on EFSA's official YouTube channel and on the EU Academy website. This webinar will refer specifically to the pest Bactericera cockerelli. Lastly, for the new attendees and as a reminder, since the microphones are mute, the Q&A chat is enabled for you to ask any questions that you may come up while the experts are presenting. Before we get into the content of the webinar, let me remind you some housekeeping rules. You are automatically connected to the audio broadcast, one-way audio, listening only mode, and participation is not permitted. The event is in English. Questions should be submitted in English via the Q&A chat. If some questions remain unanswered, you can resubmit them via email. Please consider that official answers will be published in the EU Academy only after the webinar. This event is being recorded, and recordings will be published on EFSA's official YouTube channel and on the EU Academy. We will send all the information after the webinar. After the event, attendees will be requested to evaluate this EFSA's event filling out a Microsoft survey. As you can see on the screen, this is the agenda for today. Now we are in the presentation, and in some minutes, I will hand the floor to the experts for the presentation. Before we end this webinar, we will do a short quiz, and then we will get to the end of the webinar. So who we are? Today with us is Luca Mustapik. He's an agronomist specialized in plant health and pest management, a member of the EFSA Plant Pest Surveillance Network. And today also with us is Lucia Zapala. She is full professor of general and applied entomology at the Department of Agriculture, Food and Environment at the University of Catania in Italy. My name is Jose Ovejas. I'm a journalist and I will be the moderator of this webinar. So before we get into the experts' presentation, we would like to invite you all to participate in a poll to see who is in the room. Please proceed to search www.menti.com with your mobile phone or computer. To participate, you can either use the QR code or enter the code that will be displayed on your screen in a few seconds and click the join button. Before we begin with the poll, I would like to stress that Mentimeter will be also used at the end of the webinar to display several questions that the expert have prepared to check if participants understood the contents of the webinar. Therefore, it's advisable for you to keep this in mind and have an electronic device nearby to expedite the process. So as you can see in the screen, you can scan that QR code, or you can join the menti.com with the code 85059745. Okay, I can see half of you are from the National Plant Protection Organization at central level. We can see 14 people from the National Plant Protection Organization regional level, a couple from the support laboratory diagnostic. Okay, so let's move on. And now is the time for me to hand over the floor to our 2 experts. So Luca, Lucia, the floor is yours.
Thank you, Jose, for the introduction. Hello, everyone, once again. My name is Luca. Today with me is Lucia. Hello, Lucia.
Hello, Luca. Hello, everyone attending the seminar.
Yeah, can you just confirm that you see the slides and presentation?
Yes, indeed. I can see them clearly in presentation. Thank you.
Okay, great. Then we are good to go. So this is the plan for today. We are going to be running the repest tool in order to design a detection survey focused on today's pest, which is the potato psyllid or Bactericera cockerelli. Briefly, we will remind the participants about the EFSA methodological framework for the survey design, but mainly we are going to be talking about the main parameters for the application of the framework focused explicitly for this pest, and in particular, we'll be showing how to reach a sensible epidemiological units, how to set inspection units, risk factors, detection methods, and also confidence level and design prevalence. Before we go into the scenario, really quickly, I will just remind everybody about the EFSA methodological framework. If you watched previous webinars, I'm sure that you know all about this, but in essence, the EFSA methodological framework is well embedded within the tool called Repest, currently version 3.1, and its main goal is to solve one of the main issues of the plant health surveys, which is that we cannot inspect the whole host plant population, the target population, since usually it's too large, it's practically impossible. So this tool allows us to calculate a sample size which is representative of the whole target population. And this is the scenario for today. So we are playing a role of an MPPO, which was notified recently that our garden center is importing plants for planting of tomatoes from the areas where Bactericera cockerelli is known to occur or present. And we ordered a risk assessment, and our risk assessors have notified us that this risk is actually very high, because also in our proximity of this area, we have a large potato production field of 2,900 hectares. So our main goal as the MPPO and the risk manager is to know what is the pest status in our potato fields. And let's get now right into the Repest tool to start a survey from the scratch. If you have a save session, you can always load your survey, but for this purpose, we are going to be starting a new survey from the start. We are going to be running a detection survey since the Bactericera, as we will see, is not present currently in EU. So this is a survey that we are conducting to elaborate on our pest freedom. We are in the initiation phase here. The first, we need to set the goal, and usually we are running this annual survey, so priority pests to be compliant with the regulatory requirements. Then we need to choose the pest from the drop-down menu, and if you just type the first few letters, you will easily find it. And then, as far as we know, in the EU, it's absent, and usually the surveys of all priority pests are done annually, so we can assume that the last survey was last year, and we conducted a detection survey as well as for this year. And now I think it's a good time to switch to Lucia to introduce this pest and its biology.
Okay, great. Thank you very much, Luca. So the object of today's seminar is this tiny jumping plant lice, or psyllid, whose scientific name is Bactericera cockerelli. It actually belongs to the order Hemiptera, superfamily Psylloidea, and family Triosidae, that is so called because of the trifurcate basal vein in the forewings, which is a diagnostic character for this family. The species, which is commonly known as potato or tomato psyllid, is listed as a union quarantine pest in Annex 2, Part A of the Commission Implementing Regulation 2019-2072, being it not known to occur in the EU territory, as Luca was mentioning. It is also listed as a priority pest under Commission Delegated Regulation 2019-1702, implying the obligation, as Luca was reminding, for annual surveys for this pest. Special requirements are laid down in Annex 7 on freedom from pests related to import of fruits of Solanaceae, which is a family that is highly attacked by this pest, from countries where the insect is present. So let's go see where this insect is present. Can you switch to the next slide? Thank you, Luca. So this pest, Bactericera cockerelli, is believed to be native to southern, North and Central America. It is not known, as we were saying, to occur in the EU, and its historical range and genetic diversity are heavily concentrated in the Western United States and Mexico, and through human-assisted dispersal, it has reached parts of South America, like Ecuador, and Oceania, like New Zealand and Australia. Next slide, please. By combining both empirical observation with bioclimate data, so using a correlative ecological niche model, for large climatically suitable and optimal regions that are here highlighted with dark red were identified in South America, Eurasia and North Africa, Sub-Saharan Africa, and Australasia. These areas are areas of potential invasion and establishment of Bactericera cockerelli and of the causal agent of the disease that this psyllid is able to transmit, that we will talk about in a few minutes. When making a close-up to Europe, to the EU actually, taking into account the distribution of Bactericera cockerelli in areas where it has been already reported, such as the Americas and New Zealand, it is assumed that it would be able to become permanently established outdoors in southern and central areas of the EU, as well as in northern areas that have mild winters. Actually, establishment in the east of Poland and farther north is unlikely, but transient populations could be possible. Besides, it should be mentioned and reminded to all of us that many of the psyllid solanaceous host plants are also cultivated under protected conditions, which would provide good conditions for its establishment. Finally, in terms of invasion potential, also the wide distribution in the EU of susceptible crops that we will see are mainly solanaceous crops, highlighted as well the relevance, both in terms of cultivated surface as well as amount of production, is another element that would help the establishment of this pest in the EU. Next slide, please. So let's see how does this tiny sap-sucking insect develop and behave. The adult females, they lay eggs individually on the lower or upper surface of the leaves, usually near the leaf margin, and generally on the upper part of the plant. After egg hatching, the young nymphs crawl down the egg stalk to search for a place to settle and feed. And nymphs usually remain in their place sedentary during the entire development, preferring sheltered and shaded locations. They go through 5 nymphal stages, and then female and male adults emerge, and they are ready to mate after a few days. Nymphs and adults, and that's also a characteristic feature, they produce characteristic and large quantities of small whitish excrements, which may adhere to foliage and fruit. Adults are good flyers and readily jump when disturbed. Female produce chemical cues were proved to attract males before mating. Actually, this has been proved at the experimental level, but there's no practical application of these chemical cues yet. Egg laying starts a couple of days later, and oviposition lasts around 50 days, and female lay an average of 300 up to 500 eggs over the lifetime. And actually, this lifetime total adult longevity ranges from 20 to 62 days, and females usually live 2 to 3 times as long as males, depending on the host plant that they attack. Weather is an important element for all insects, as we know, but in particular for these kind of insects, and it's influencing the biology of Bactericera cockerelli and its potential damage to host plants. It seems to be adapted for warm, but not for hot weather. Cool weather during migrations, or at least the absence of high temperature, has been associated with several insect outbreaks. Actually, optimum psyllid development occurs at approximately 27 degrees, whereas oviposition, hatching, and survival are reduced when temperature go upon 30 degrees, 32 degrees, and stop totally at 35 degrees Celsius. Next slide, please. So let's see its life cycle. So the potato psyllid actually can complete multiple generations per year, considering that under optimal temperature conditions that we said are around 27 degrees, the entire life cycle lasts around 2 weeks. So this makes it able to complete lots of generation during the year. Thinking in terms of surveillance, survey timing depends on the host plants and local environmental conditions, of course. But surveys should be started when the target pest is active and condition support is present, which varies with microclimate in each member state. It can be all year round in hottest Mediterranean areas or during summer in other areas. Visual examination and trapping actually of Bactericera cockerelli, as we will see later on, and other psyllids also, should start at the beginning of the cultivation of the growing season and continue throughout the life cycle, the production cycle. In general, the best period, and we will see it more in details, to observe adults is from April to October. Generations often overlap. This complicates the evaluation of population dynamics during outbreaks. And the potato psyllid overwinters as adult on evergreen or perennial plants in the Solanaceous family, such as bittersweet nightshade or matrimony vine, or both cultivated and uncultivated non-crop hosts that serve as a critical bridging reservoir where the psyllid survives harsh winter conditions before migrating to infest seasonal cash crops in the spring. The potato psyllids can survive cold winters. There are reports of nymphs and adults surviving at -15 or -10 degrees, respectively. Besides, a recent research proved the higher cold tolerance and therefore overwintering success of potato psyllids infected with the bacterium that we will talk about in a very short while. Next slide, please. Indeed, as anticipated, this psyllid is able to transmit the bacterium Candidatus liberibacter solanacearum, which is a phloem-limited, gram-negative, unculturable, or fastidious, as it is called, bacterium, associated with several emerging diseases. Among them, it is associated with a potato disease, which is called zebra chips, that causes significant economic losses by reducing both yield and quality of potato crops, as well as other diseases in other solanaceous crops and other also crops. Several haplotypes, actually, of these bacteria have been identified with different letters from A, B, C, D, E, F, U, G, and 2 different H haplotypes, and CRAS1 and CRAS2. Bactericera cockerelli is known to transmit haplotypes A, B, and presumably F. Actually, some of these haplotypes, as you can see in the slide, were reported as being present in the EU. These are not the ones transmitted by Bactericera cockerelli, they're transmitted by other psyllids, and they are mostly noxious, they damage apiaceous plants.
Okay, Lucia, thanks. So I'm thinking here from the map, it looks quite complex. There are many haplotypes of the disease, but basically, if we see the symptoms of disease in our potatoes, there is a high chance that the Bactericera was the vector. So, because as I understand, no other vectors in Europe transmits the disease into the potatoes, right?
Yes, thank you, Luca, for your very good question. And actually, you're totally right. No other vectors are known to transmit these haplotypes related to solanaceous crops, and in particular potato. Actually, in solanaceous plants, so this bacterium is primarily spread from insect from infected to healthy plants by these psyllid species. However, the bacterium can also be transmitted by grafting and via dodder, which are parasitic plants of the genus Cuscuta. And besides, the bacterium can also be transmitted through seed potato tubers. Not through true seed or seed from other solanaceous plants. However, the risk of Candidatus liberibacter solanacearum transmission by tubers is very weak. So it can spread by the movement of plants for planting that are infected, by grafting of young tomato and pepper plants, but basically on potato, the main transmitter, the main vector is the psyllid, although potentially also seed potato tubers can transmit the disease.
Okay, great.
Okay, so continuing a bit, as we understand that these two biomes are strongly connected, these bacteria is transmitted by the psyllid insect vector to new host plants. In addition, as I said, it can be also transmitted by propagative plant material or parasitic plants. The bacterium is found in several organs and tissues of the psyllid host, including the alimentary canal, salivary glands, hemolymph, and bacteriomes that are specialized organs to contain bacteria, and is transmitted in a propagative, circulative, and persistent manner. When a psyllid feeds on the phloem sap of a plant infected by the bacteria can be ingested by the insect. This time is called acquisition access period, and the bacterial cells must pass through the alimentary canal wall, move through the hemolymph, and finally reach the salivary glands. The bacteria can then be transmitted with salivary secretion into a new host plant during psyllid feeding. The latent period, which is the time between the pathogen acquisition and the potential for the psyllid to transmit it to a new plant, is at least 14 days. And it has also been seen that although newly emerged potato psyllid adults efficiently acquire the bacterium within 24 hours, 48 hours, transmission may remain low at 14 days post-acquisition due to low bacterial load. So it has to increase its load to efficiently, more effectively transmit the bacterium. Besides, vertical, so transovarial transmission of this bacterium has been reported in Bactericera cockerelli. Therefore, the bacteria can also be detected in the eggs, nymphs, and then the adults of the progeny of infected female psyllid. When the concentration of the bacteria I'd like to spot is low, plants may remain asymptomatic. And then we can see where the bacterium is present. Next slide, please.
Before we go, I'm just wondering, maybe you already said this information, but maybe it's good to repeat. How much is the inoculation period, meaning, and the asymptomatic period? So can we, let's say, if a healthy plant is infected in the same season, can we see the symptoms in the host plants during the same season? How long is this period?
Yes, we can definitely see the symptoms in the same growing season, definitely.
Okay, that's good.
Thank you. So, as we said, Candidatus liberibacter solanacearum is the complex of all these the haplotypes that we mentioned. So, if we see spots in Europe, in the EU, these are all related to other haplotypes. It is included in the list of regulated non-quarantine pests. And Annex 4, Part G of Commission Implementing Regulation 2019-2072 on seed potatoes. And haplotypes A, B, and presumably F that are associated to solanaceous species are not known to be present in the EU. And
okay,
then
thanks. Thank you, Lucia. Then we will go back now to the repest a bit to go further into design. Now we know a bit more about the pest itself. I think the first relevant information that you presented here is that basically the whole EU territory could be suitable for the establishment of this pest. And since we are using our imagined country of survey land. I think all of our area is also suitable for the establishment. So I'm going to choose the whole survey area, the whole country as a survey area. And then what I'm wondering is, you mentioned that the host plants of this pest are the solanaceous crops, but I'm wondering what would you recommend for the detection surveys?
Okay. Yes, actually, the species is listed as being polyphagous since adults have been collected on a wide variety of plant families. However, eggs are laid and nymphs feed and develop only on a few plant species of the families Solanaceous, Convolvulaceous, and Lamiacea. So definitely Solanaceous cultivated crops such as potato, tomato, sweet pepper, and eggplant would be the most relevant for the psyllid to lay eggs and to reproduce. So certainly it can be considered that it has a clear preference for tomato, for potato, and for eggplant, sweet pepper, and also solanaceous weeds are also reproductive hosts, and this is very relevant. Such as, for example, the perennial plant that I mentioned before, the bittersweet nightshade, which is widely distributed in Europe and is a year-round host of the potato psyllid. Considering both the bacterium, but especially the psyllid host range, when surveying for Candidatus liberibacter haplotypes that are related to solanaceous crops and their vector, Bactericera cockerelli, surveillance should be carried out on solanaceous species, and in particular, I would say on tomato and potato for sure.
Okay, so we will be, I think, focusing on these two crops, but you are mentioning also some wild solanaceous crops. I think those could be also relevant in case of, let's say, a delimiting survey, because it could be a reservoir for the pest in the overwintering stage. But in any case, I think for our case, we are safe to focus on tomatoes and potatoes. Okay. Then we will choose potatoes, Solanum tuberosum and Solanum lycopersicum. Then we go further. We do not have a shapefile with our host plant distribution, and we are now in the preparation phase. What is the next step in the survey design? Here you can always consult the pest survey card of EFSA on Bactericera cockerelli. The link is always provided here. And our now main objectives are to characterize the target population. So we are starting with the target population. For this, I will go back to the slides a bit. Very briefly, we will mention the structure of the target population. As you already know, it's a survey area divided into epidemiological units. Within these units, we survey the inspection unit depending on the methods we are using. And within these inspection units and the target population as a whole, we want to introduce usually a risk factor, since we want the survey to be risk-based. Starting from the level 1, as I already showed, the survey area will most usually depend on the climatic suitability and potential of establishment of the pest. As we already showed, the whole survey area is then chosen. And for the epi units, we, beside the major potato production, which is in the vicinity of our risk location as per our scenario, we also have in another region on the right, a major open field tomato production. And as a risk manager, we decided that we want to focus on these two most economically important crops for us. Next step we need to set is the inspection unit, and it's the unit which is examined for the pest present presence. It can be trap area, plant, or even a pest vector, as in our case. In any case, for our scenario, we decided that we are going to be using trapping as our main method combined with a visual examination. So our inspection unit is the area covered by a single trap. So let's go first to the inspection unit calculation, since this is the first, let's say, quantification that we need to provide to the tool. In order to calculate the sample size, we need to know how many inspection units we have in our area. As we already mentioned, our inspection unit is the area covered by a trap. And we need to calculate how many of these cells we would need to have to cover with our traps. And you can do it. We are first calculating if we have 2,900 hectares. If we divide it via the trap coverage, we get around 288,000 cells of this area of 100 meter squares, which is covered by a single trap. And this number of cells we would need to cover the whole potato field production. Similarly, you can also arrive at the number of plants per hectare. This number is needed in case you want your inspection unit or your detection method to be visual examination only. And similarly for tomatoes, you can get your plausible figures of tomato production. And once you have a between row and between plants distance, you can calculate how many plants you have per hectare. And then if you have 2,900 hectares, then we would get around 140,000 cells of this trap area and to cover the whole tomato production. So this is the first two parameters that we covered, and now we are ready to go back to RIPES to carry on. As we mentioned, the inspection unit in our case is the area covered by a trap, and we said that we want to combine multiple field detection methods that use different types of inspection units. This is exactly our case. And then we see that we have two tabs here with the two host plant categories that we've chosen. So let's go first with the potatoes. We need to answer, can the target population be infinite? We click no, because we just show that we know exactly how many cells our target population contains. And for the epidemiological unit, we can choose the user-defined option to describe our epidemiological unit as potato open field production. And very importantly here, the number of inspection units is the number of cells. So we enter the number of 288,000 cells. Okay, this is then set for the potatoes. We do the same for the tomatoes. Again, the target population is not infinite. Epidemiological unit is 1, which is we are considering the tomato open field production. And again, we had 140,000 cells to be covered by the trap. Now, once we are finished with the inspection unit, the next parameter are the risk factors. For sure, we want to conduct a risk-based survey, but we still don't have the information on this, so I will go back to the slides to show you what we need to do in order to set our risk factor. By the definition, as you know, it's a biotic or abiotic factor that increases the probability of pest presence. In our case, there are multiple risk factors that we could consider. So I'm going to ask Lucia to recommend some.
Yes, well, I think that although, as you were saying, there are several pathways for Bactericera cockerelli introduction, the main risk factor would be trade of solanaceous plants for planting from areas where Candidatus liberibacter, solanacearum, and especially Bactericera cockerelli are known to occur. And indeed, if we look a bit more into its dispersal, certainly there is natural dispersal since the adults, as we were saying, are able to move and they are actually able to cover long distance and actually maximum distance expected to be covered in 1 year by Bactericera cockerelli is 100 kilometers. Other pathways of introduction are human-assisted pathways, which are seed potato tubers and where potato tubers, if tubers have sprouts or green parts that are needed for the psyllid to reproduce. Fruits of solanaceous with green parts, such as truss tomato or sweet pepper with peduncle. Other living parts of solanaceous, such as cut flowers, cut branches of some ornamental physalis, for example, plants in the families of convolvulaceae, and such as the sweet potato or lamiaceae, such as mint, but definitely the more relevant pathway, the major pathway can be considered plants for planting of solanaceae other than potato, which are covered by seed potato tubers.
Mm-hmm. Then what you would recommend as a risk factor?
So definitely import and trade of solanaceous plants for planting, which are pepper, sweet pepper, aubergine, tomato, and potato from areas where the bacteria and the psyllids are present would be recommended as risk factors.
Mm-hmm. Okay, all clear, thanks. And then for risk assessment framework, we've given here just a couple of risk activities that could be considered in case of this pest and its dedicated risk locations, risk areas, the relative risk, and the justification. As Lucia mentioned, movement of infested fruits could be also relevant. Import of plant parts, such as cut flowers from infested areas, is also a medium relative risk, but for our case, we'll be considering a risk factor related to the proximity to our garden center, which is a risk location, and in particular, the distance of the risk location to the potato production. So each potato production field less than 1 kilometer from the garden center is our highest relative risk. And this is the structure of the target population that is fully set for our scenario. And as we mentioned, for the risk factor, the risk factor is the proximity to a risk location. The activity is the production of potatoes in the proximity. The location itself is the fields, and area at risk is, as we mentioned, the surrounding of these risk locations. Now, once we have highlighted our risk factor, let's go back and enter this information into the repest. So the risk does not, in this case, depend on the spread capacity, but it depends on the proximity to the risk location. So we will be using only one risk factor. We can label it just with a risk factor 1, and shortly we can describe it as the production of potato field, of potato open field in proximity to the garden center. This is just to be used in the report later. And now we have our risk-based survey ready for the potatoes. Next, we need to characterize the survey sites and detection and laboratory methods. For this, we'll be showing next few slides. Lucia, can you take over for this?
Yes, of course. Thank you, Luca. Yes, definitely. For surveying, we reminded before that Bactericera cockerelli can be observed and identified visually on plants by visual examination, collected with plant samples. Indeed, Bactericera cockerelli Adults can be hand collected, and of course, collection of samples of egg and nymphs require visual examination of the foliage of these plants. Adults can be trapped effectively using yellow sticky traps, and we will see more details on timing and density of traps in the next slides. Thank you, Luca. Yes, so as already said, survey timing depends on the host plant and of course on the local environmental conditions. Traps should be placed in the field when the target pest is active and conditions support its presence, which varies with microclimate in each member state. It can be all year round, as we said, in hottest areas or during summer in other areas. Visual examination and trapping of Bactericera cockerelli should start at the beginning of the potato season and continue for at least 10-12 weeks. In general, the best period to observe adults is from April to October. And if we look at the production timeline of Bactericera cockerelli main host plants that you see there displayed on the slide, tomato, potato, and sweet pepper, this corresponds to the time interval between transplanting and harvesting. Without forgetting that generations often overlap, so this complicates a bit the evaluation of population dynamics during outbreaks. So psyllid abundance, also we have to remember that early in the season is more concentrated, is more abundant on the field hedges, and as the season progresses, it is more evenly distributed throughout the fields. So if we want to have more details, can you switch to the next slide, please, Luca, on the traps and how to place them? It has been seen and proved that yellow sticky traps or yellow water traps, but mostly yellow sticky traps are used preferentially to trap adult psyllid. It's also true that using sweep nets and vacuum trapping or sampling leaves, so visual examination and sampling, may also be used, but these methods are less efficient. It's important to set the traps low, just below the top of the plant canopy, and this is the position that seems to give the better results. And then they need to be put higher up with the growing crop, and they should be replaced weekly or every 2 weeks, depending on the season. Egg and nymphal sampling, of course, cannot be trapped cannot be done with the traps, but can be done only with visual examination of foliage, remembering that populations are initially highest at the field edges, and then they become more evenly distributed. For crops grown under protection, so in greenhouses, traps may be also placed near potential points of entry. A recent developed 3D printed trap allows to collect adults and directly put them into preservative, which is 70% ethanol. And this greatly facilitates, of course, molecular testing, especially for the bacteria infections. Although it can be said that if the insects are collected from sticky traps, it is not needed to remove the glue before a DNA extraction, and bacterium can be reliably detected in infected psyllids for up to 10 months on sticky traps stored at room temperature, of course, inside. For long-term storage, of course, psyllids before testing should be preserved in 70% ethanol. Next slide, please. So since traps should be gradually moved, as we said, up as plants grow, it is recommended, or it's easier to attach them to wooden stakes so they can be easily put higher. One trap, as Luca already anticipated, covers around 100 square meters of potato, tomato in open field. From 10 to 30 traps per hectare should be placed. Consider that placing a higher number of sticky traps in a field increases, of course, the likelihood of early psyllid detection. In greenhouse, one trap per 300 square meters can be considered together with traps at the entry point. Traps, of course, should be adequately protected, packed, and allowed to collect adults and then proceed to identification and even bacteria detection. next slide, please, to see some of the symptoms of these psyllid. And it's actually not easy to distinguish symptoms caused by the plant pathogenic bacteria and those caused by its vector. Symptoms caused by the psyllid are called psyllid yellows and are related to the psyllid feeding activity and injection of toxic saliva. On the above ground plant parts of potatoes and tomatoes, Bactericera cockerelli may cause characteristic uprightness of new leaves, retardation of growth, chlorosis, purpling of leaves, thickening of terminal internodes, enlarged nodes, and also aerial potato tubers, as can be seen in the slide. Plus, below ground, the psyllid may cause excessive numbers of very small potato tubers, very small misshapen potato tubers. Another characteristic symptom connected to the presence of the psyllid is, as I said, the presence of sugary excrements that are called honeydew, and that can look like whitish chalky particles. If we switch to the next slide, we can see some symptoms instead of the caused by the bacteria. On potato plants, symptoms are described as being similar to the psyllid yellows, making it difficult to distinguish the symptoms, including chlorosis, zigzag-shaped stems, axillary bud proliferation, swollen nodes, as I was saying, vascular discoloration, wilting of plants. And then there are also the underground symptoms, smaller tubers, increased in number of tubers, as I was saying. But mostly, the most important symptom is browning in medullary ray tissue throughout the tuber, which is, as shown in the picture, more evident in fried potatoes. And this is the reason why the bacteria, the disease is called zebra chips. Okay, I think this is enough for symptoms. Thank you, Luca.
Thanks, Lucia. And the last information that RIPEST will ask us is how many potato plants will be visual examining while we are going to put our yellow sticky traps in the field. And I found one reference for this information. In our case, usually I think during the visual examination, our inspectors inspect around 20 nearby plants when they switch also the traps. So usually in the smaller plots, such as 1 hectare to 5 hectares, it's recommended to visual examine about 20 up to 100 plants. And then the last thing we need for these parameters is the method sensitivity, which is the combination of sampling effectiveness and diagnosis sensitivity. We are using the values from the recent ECE on this pest, which will be published, I believe, tomorrow or today. And you can see that the effectiveness of the visual examination is quite lower than the effectiveness of the insect traps, which we are using the yellow sticky traps. In any case, we combine it with the two available methods, which is the morphological ID and the molecular available methods, and we are deeming them highly effective. So the overall method sensitivity reaches around 80%. Speaking about the laboratory identification, diagnostic characters of the potato psyllid include, of course, a white band on the dorsum of the first abdominal segment and the V-shaped white mark on the last abdominal segment. Of course, all of these morphological characteristics are available in the keys, and you can use it via, you can see it via the light microscopy. Again, for the nymphal instars, they are not usually used in the morphological identification in the laboratory. They are quite small, as opposed to the adults that measure about 2 to up to 3, let's say, millimeters in length. And as Lucia already mentioned, a real-time PCR is also available in case the morphological ID is not conclusive. And lastly, here is the overview of all detection and diagnostic methods available for this pest. Usually, yeah, in the field, you can combine yellow sticky traps or trapping with the visual examination. In any case, if you collect the pest, it should be an adult, and those adults should be preserved in the alcohol, ethanol 70%, and sent to the lab where you can use morphological molecular methods. The good news is that we have both the EPO protocol and the diagnostic protocol from ISPM for this pest and the CCO, which is the disease. We are now finished with this part of the parameter. Now we can go back to the repest once again. I just need to correct a bit here because we do not want a risk-based survey for the tomatoes, only for the potatoes. So that's fine. Now the type of the survey site is going to be our open field production. The survey period is already highlighted, as Lucia mentioned, usually from the July up to the October is the preferred timing. And then for the detection identification methods, we are going to be using two different methods. As we said, the first one, the primary, is the insect traps, sticky yellow traps, which are going to collect the pest. And as we know, the area covered by single trap is 100 meters square, which is in hectares 0.01 hectare. In the average number of host plants per hectare, as we already shown, are around 44,000 plants per hectare. This is also important for the tool. And then we do the same for the different field, the second field method, which is the visual examination. As we shown, usually around 20 plants we inspect in each inspection unit. We chose the visual examination as a method. We can focus on both the pest, we can look for the pest and the symptoms on plant. Usually we want to collect the adults. So the sampling matrix is the pest itself, and a really nice thing is that the repest already gives us the sampling effectiveness of each method. And now we need to do the same for the potato. So again, just repeat the whole process. We enter the needed information. So field method one is the insect traps, the sticky traps. It's collecting the pest. Its coverage is 0.01 hectare. The average number of tomatoes per 1 hectare is around 22,000. And then for the field, let's not forget the second method. Again, around 20 plants we will be visual examining. When we are going to be putting our traps, all the same information as for the potatoes. And now, once we are finished, the last thing is the laboratory, describe the laboratory methods. Usually it contains the isolation up to the confirmation. We said that you can use both morphological and molecular ID, and our diagnostic sensitivity is usually quite large. Now we are up to the final part of the design, which is to just set the confidence level and design prevalence. For this purpose, I will go back here because we also need to calculate what's the proportion of high-risk target population. Since we have said that our high-risk area is 200 meters from the risk location, which is the green band. And if you calculate the size, it's 12.5 hectares. And our approximation is that relative risk in this area is 5. So 5 times more than the baseline, which is the rest of the potato production. And notice that we are only considering the potatoes as risk factors and not tomatoes. Once we have our relative risk levels, the last two parameters are the confidence level and design prevalence. These two parameters often are most detrimental to the sample size. Design prevalence is the maximum prevalence of the pest that could go undetected. Usually, and the confidence level represents a reliability of our survey results. In essence, the goal of each of our pest freedom surveys is to conclude with a high enough confidence level that our pest is below a certain maximum prevalence or this design prevalence threshold. And finally, we have all the parameters set and explained in order to RIPEST to successfully calculate our sample size. So last two parameters are going to be confidence level of 95% and design prevalence of 1%. So you can see already in the RIPEST, it's preset to 95%. Let's first calculate the sample size for the potatoes. You can see design prevalence is already set to 1%. The tool asks us how many risk levels we want to take into account. As we show, our high risk area is the relative risk of 5, and it's only 0.4% of our whole target population, and the rest is the baseline. So we can click now simulate, and we finally arrive at our theoretical sample size, which is 21 traps in the high risk area and 107 traps in the baseline. We do the same for Solanum lycopersicum, but without the risk factor. And for some reason, it's not working now, but in any case, we can continue. We do not want to reallocate the samples to epi units and also to the geographical coordinates, and we arrive at the conclusion. This is where you can enter your results when you get the results from the laboratory and you finish your annual survey. You can here put if you had or if you did not find the pest, you can put here your results. They don't have to be identical to the theoretical sample size. For example, you maybe only could collect or put 20 traps in the high risk area and 100 traps in the baseline. In any case, now this allows us to calculate the achieved confidence level, and it's divided into high risk area and baseline. And similarly, you can do it for the tomatoes, and then you would reach the overall conclusion of your confidence level achieved for the whole survey. And then the last step, you can download your report in the PDF, or also you can download it in the Excel file for your Eurofit reports. And now this is the last slide of our presentation. It shows our calculation. So you can see for the potato production, the tool gave us a number of around 20 traps to put in the high risk area and about 107 traps to put in the baseline. So to cover all other potato field area, as opposed to the tomato, where we did not consider a risk factor, and there we calculated that we would need around 210 traps to put in our tomato production in 1 year. You can see there the overall conclusion we reached is that we are slightly above 95% confident that our pest is below a 1% design prevalence. With that, I thank you all for the presentation, for the attention, and switch back to Jose.
Thank you, Luca. Thank you, Lucia, for your presentation. Now is the time to do a small quiz to test yourself and see if you've understood the webinar contents. So for this, we need to use Mentimeter again. Please take your device and scan the QR code that you will see in some seconds on your screen, or type www.menti.com on your browser and introduce the code that you will see at the top of the slide. As you can see now the QR code, you can scan it or just type www.menti.com and use the code 5773 5379. Okay, so let's go for the first question. What is the main plant health concern associated with Bactericera cockerelli? Option A, direct feeding damage. Option B, transmission of Candidatus liveribacter solanacearum. Option C, root knot nematode transmission. Option D, potato virus Y transmission. Okay, so most of you think the correct answer is the option B. Let's move on to the second question. Which type of trap is most commonly used for the detection of adult Bactericera cockerelli in potato fields? Option A, pitfall traps. Option B, light traps. Option C, water traps. Option D, yellow sticky traps. Okay, so all of you think the correct answer is the option D. We'll see after the test. Okay, let's move on to the third question. If we choose the inspection unit for Bactericera cockerelli detection survey as an area covered by a trap, what is the respective target population? Option A, total number of traps in the survey area. Option B, total area of host plant production to be covered by a trap. Option C, total area of host plant production in hectares. Option D, total number of host plants in survey area. Okay, this one is not, you're not sure as the last one. So let's move on to the fourth question and we'll see the correct answer after. Okay, which is the major pathway for Bactericera cockerelli introduction in the EU? Option A, potato towers. Option B, wood packaging material. Option C, plants for planting of Solanaceae. Option D, natural spread. So let's move on to the fifth question. And the last one. Which of the following statements is true? Option A, Bactericera cockerelli performs one generation per year. Option B, Bactericera cockerelli develops uncultivated and wild Solanaceae species. Option C, Bactericera cockerelli transmits all known Candidatus liveribacter solanacearum haplotypes. Option D, none of the above. Okay, so most of you think the correct answer is the option B. We'll see now with the experts. So Luca and Lucia, if you can give us the correct answer, please.
Yeah, will I start, Lucia, or you want?
I may start and then maybe you can pop in. Okay.
Yeah.
So the first question was, what is the main plant health concern associated? And I think that most of the people answered correctly, which is the transmission of the bacterium that Bactericera cockerelli is able to transmit. The direct feeding damage is relevant, but not as relevant. So the option B was the correct one. The second question was which type of traps is most commonly used? Actually, the best option and the best choice are yellow sticky traps, as we saw. So option D was the correct answer. And then I may leave the ground to you, Luca, for these on the...
Yeah, for the question 3, if we choose the inspection unit for Bactericera as area covered by a trap, our total target population should be a total area of host plant production to be covered by a trap, which is the survey cells, as opposed to, let's say, if we would consider an inspection unit as an individual host plant, then we would need to calculate a total number of plants in an area. The fourth question, which is a major pathway for Bactericera cockerelli introduction into EU, there are many, but we consider that plants for planting of Solanaceae is the one which poses the highest risk. And then the last question, which of the following statements is true? The option B is correct, which is that Bactericera cockerelli develops on the cultivated and both wild Solanaceae species. So don't forget that also these wild weed Solanaceae species could be an important reservoir and where the pest could go undetected.
Thank you again for your presentation, Luca and Lucia. Now we are getting to the end. Thank you all for attending today. In case we did not manage to answer all the questions, they will be published on the EU Academy. Please note that similar questions will be grouped where possible. The recordings of today's event will be available on EFSA's official YouTube channel and on the EU Academy with subtitles in 4 different languages: English, Spanish, French, and Arabic. Next webinars will take place on Butzaphelen-2 Xylophilus in September and Philosticta citricarpa in October. So before we leave, please take a few minutes to scan the QR code on the right side of the slide and fill in the evaluation survey. If you are unable to scan the QR code, after the event, you will receive a link to access the Microsoft survey form. Your feedback is essential to improve our future events. So thank you everyone for attending this webinar, and thank you once again, Luca and Lucia, for the presentation. We have now reached the end of the session. I hope you have all learned about this pest, and you are now well informed. As I mentioned in the previous slide, the next webinar is scheduled for September. I hope you can all attend. I wish you all a great summer. See you.